Clicker questions for Chapter 8, Protein Synthesis, Processing, and Regulation. Number 1, if all tRNAs have the same CCA nucleotide sequence at the 3' terminus, amino acids are covalently attached to the ribose of the terminal adenosine, how can a specific tRNA recognize a specific amino acid? Letter A, the 20 individual amino acids recognize sequences along their specific tRNA. B, the 20 amino acyl tRNA synthetases are specific for each amino acid tRNA pair. Or letter C, the tRNAs use complement base pairing to fold around the specific amino acid. The answer is B. The 20 amino acyl tRNA synthetases are specific for each amino acid tRNA pair. Now, it does allow a little bit of relaxed base pairing, which can also be termed as non-standard base pairing or a wobble, where the last portion of the base pair, where the guanine and uracil base pairs can partly bind because of either the guanosine or inosine binding the anticodons of several tRNAs during the processing. So they can bind to either uracil, cytosine, or adenine in the third position. Number 2, what do ribosomes have in common with RNase P and self-splicing introns? A, all have innate ribosome activity. B, all are composed of protein RNA. C, all are found only in eukaryotes. And D, all are regulated by protein modification. The answer is A, all have innate ribosome activity. It's technically the large subunit that allows the ribosome activity. It allows cleavage when the final mRNA strand is terminated and separated from the large subunit of the ribosome itself. The other answers here, all are composed of protein RNA. Very simply, self-splicing introns will not have the protein themselves. Letter C, all are found only in eukaryotes. Anytime you see a statement like that where it's very binary, it's either only or not or always or never, most oftentimes there'll be a false statement. Letter D, all regulated by protein modification. Again, self-splicing introns, since they're self-splicing, they don't need a protein to aid in the splicing. Number 3, mitochondrial and chloroplast ribosomes resemble bacterial ribosomes. This is because A, both are eukaryotic organelles, or B, both are semi-autonomous organelles, C, both organelles arose endosymbiotically, or D, chloroplasts arose from mitochondria. The answer is C. Both organelles arose endosymbiotically. This is actually borrowed from Biology 1, where we talk about how in two separate aspects of evolution, mitochondria and chloroplasts most likely came from prokaryotes that invaded a bacterial cell and it basically achieved a symbiotic relationship over time. For animal cells, of course, it was only mitochondria, and of course for plant cells, it was a combination of mitochondria and a second vagination of another prokaryote that did chloroplastic activity. And since then, we have the eukaryotic cell. Prokaryotic ribosomes initiate translation by recognizing a specific nucleotide sequence in the mRNA, whereas eukaryotic ribosomes recognize the 5' mRNA and cap. What is the basis for this difference? Is it letter A, eukaryotic genes are monocistronic, prokaryotic genes are polycistronic? Letter B, prokaryotic chromosomes are largely devoid of accessory proteins, like the eukaryotic histones? Or C, being polycistronic, most prokaryotic genes lack a 5' end cap to recognize? Or D, eukaryotic genes are subject to varying degrees of alternative splicing? In this case, we have two answers here. We have letter A, eukaryotic genes are monocistronic, prokaryotic genes are polycistronic. Basically mean that eukaryotic genes are monocistronic, so there'll be one particular mRNA that will be responsible for one type of polypeptide amino acid chain, whereas the prokaryotic genes, as polycistronic, will have multiple types of proteins that can be derived off one mRNA strain. Letter C also is true. So being polycistronic, most prokaryotic genes lack a 5' end cap to recognize. Of course, we know that mRNA found in eukaryotic cells will always have the 5' end cap as well as the 3' poly A tail. 5, what role does the ribosome decoding center play? Letter A, the decoding center corrects mismatches between the codon and the anticodon. Letter B, the decoding center enzymatically replaces incorrect amino acids with proper ones. Letter C, the decoding center labels amino acids for later removal by editing enzymes. Or D, the decoding center helps the proper mRNA bind to the ribosomal small subunit. Here the answer is A, the decoding center corrects mismatches between the codon and the anticodon. So when the appropriate anticodon is going to be bound onto the ribosome, onto the A site, very simply the elongation factor will allow the hydrolysis of GTP, and basically allows the next peptide bond to be chained up from amino acid to amino acid. 6. According to the textbook, regulatory proteins that bind to the 3' untranslated regions of mRNAs are also responsible for localizing mRNAs to specific regions of cells. This would be especially important in A, localizing rRNAs and tRNAs to the nucleus, B, regulating mRNA degradation and turnover, C, guiding the proper aminoacyl tRNA to the ribosome, or D, establishing cell polarity in developing embryos. Here the answer is D, establishing cell polarity in developing embryos. Now with this question, there are a couple things to unpack. This is where I'm going to get on my soapbox about Oxford Insight. These questions are derived from Oxford Insight. Some of them are preset. Some of them are suggested for use within the classroom. This is one of those things that I would actually show in the class just to illustrate a point. In the textbook, it lists one single line about developing embryos as far as actual regulation of gene translation in a particular cell. But I do want to just point out one particular term here, which I think will be very helpful for most of you, is the cell polarity. So very simply, when you're trying to target mRNAs to specific regions of cells, we'll get into this more so we get into different regions of the nucleus as well. Cell polarity very simply means where you are shunting different types of mRNAs and or proteins to a certain section of the cell. So in this case for embryos, they kind of lie in waiting until they get the appropriate signals to either further translate the mRNAs or develop any kind of protein folding and so on and so forth to basically mature the eventual proteins to be either used within the cell or shipped elsewhere. So most likely you will not see this particular question on an exam. I think this is a little bit too specific. I don't quite get into that kind of depth. I feel if they dedicate at least a few lines or at least a paragraph to a particular topic, then I'll certainly put a question on it. But something like this here, let's just use it as a teaching tool and not necessarily something I need you to really memorize as far as the next exam. Okay, number 7 is a true-false question. Chaperones are non-enzymatic catalysts. A, true, they help proteins fold, but do not catalyze a chemical reaction. Or letter B, false, any protein that is a catalyst must be an enzyme. And this answer is simply true. They help proteins fold, but do not catalyze a chemical reaction. So very simply, the chaperones just basically give a more hospitable environment to help with the folding of the particular proteins, whereas it would be less efficient if it was found in the cytoplasm, whereas they can be targeted by different types of proteasomes or other enzymes that may actually denigrate the actual protein itself. 8, misfolded proteins can be toxic in both soluble forms and insoluble or aggregate forms. True or false? And this is true. So as we looked at different types of diseases, we looked at different amyloids or tau proteins found in Alzheimer's, as well as post-CTE conditions. We oftentimes see there's no proteins that will basically be found in different types of cells that will at the very least disrupt cellular functions or at worst actually disrupt the cell and cause the cell to die. Number 9, protein maturation is A, the secretion of proteins from cell to extracellular space, B, a range of processes that alter or modify protein after translation, or C, a process of combining multiple individual proteins into a larger active complex, or D, the manner in which scaffolding proteins help nascent proteins fold correctly. Here the answer is B. So very simply, protein maturation is the range of processes that alter or modify protein after translation until its final form. 10. What would happen if an enzyme that cleaves ethanolamine were added to cells in culture that were expressing exterior proteins with a GPI anchor? Letter A, the cells will cease growth due to loss of membrane permeability. Letter B, the phosphatidylinositol portion of the anchor will be degraded. Letter C, the process of adding lipids to proteins for export will be inhibited. Or letter D, the protein portion will be released from the surface of the membrane. Here the answer is D, the protein portion will be released from the surface of the membrane. If you remember the figure that we had seen towards the tail end of this third section of chapter 8, we had a protein that was going to be attached by the C-terminus to ethanolamine to the glycolipid, and the lipid being in the inositol, which is going to be anchored within the plasma membrane. So very simply, if we cleave the ethynylamine, it would basically let the protein be free floating away from the source membrane, and therefore would be released. Number 11, a key feature of GTP-mediated protein activation is that A, GTP is permanently bound to the protein and requires protein degradation to be inactivated. Letter B, the binding of GTP initiates a dimerization activation of the ROS proteins. Or C, the GTP will rapidly hydrolyze to GDP and a phosphate, providing a built-in off-on switch. The answer is C. The GTP will rapidly hydrolyze to GDP and phosphate, providing a built-in off switch for different types of proteins. Best example that I can present to you is with the growth factors with EI F2 proteins. When they're hydrolyzed, it'll allow the binding of methionine, to be bound to the large subunit of the ribosomes in order to initiate mRNA translation. In the absence of the GDP to be phosphorylated back to a GTP, basically will shut off this particular protein and therefore will stop any further translation of a particular mRNA template. Number 12, what role does water play in the dephosphorylation of proteins? Letter A, water participates as a reactant in the hydrolysis reaction. Letter B, water stabilizes the dephosphorylated form until it can be reactivated by a kinase. Letter C, water stabilizes the pH surrounding the protein to be dephosphorylated. Or letter D, water senses the environment and controls the timing of dephosphorylation. Here the answer is A. Water participates as a reactant in hydrolysis reaction. So hydrolysis very simply means using water to break particular bonds. So A would be the best answer for this. Water does not stabilize the dephosphorylated form, and neither does water stabilize the pH surrounding the protein to be dephosphorylated. That's not its primary function. And of course, water doesn't sense anything. It's just part of the environment. Number 13, eukaryotic proteasomes are found in both the cytoplasm and the nucleus. What would be a major role of nuclear proteasomes? Letter A, degrading the introns that are removed during mRNA splicing. Letter B, degrading transcription factors in order to regulate gene expression. Letter C, degrading the nucleolus after each round of cell division. Or D, degrading and regulating DNA and RNA polymerases. The answer is B. So proteasome, especially in the nucleus, is going to help break down transcription factors, and this helps regulate gene expression. Following number 14, what happens to the amino acids that are liberated as a consequence of the ubiquitin proteasome pathway? Letter A, they be reused in the synthesis of new proteins. Letter B, they be further degraded and respired. Letter C, they're usually secreted to the external environment. Or D, they be stored in the kidneys for later use. Here the answer is twofold. A, they be reused in the synthesis of new proteins. And letter B, they be further degraded and respired. Very simply, the other two answers here, C and D. When's the last time you secreted any kind of amino acid out of anything? They usually not secreted in the external environment, usually be used within the cell to be recycled. And of course, they are simply not stored in the kidneys for later